Welcome to this lecture on arteriolar vasoconstriction and vasodilation. After this lecture, you'll be able to describe the factors which induce vasodilation in arteries, and you will be able to describe the factors which induce vasoconstriction in arteries. Firstly, we will discuss intrinsic factors. Intrinsic factors originate within the tissue and regulates the distribution of cardiac output throughout the systemic circuit locally. And this is usually done by arterioles, which change in diameter and control the amount of blood that is being perfused in a part of your body. Intrinsic factors undergo something called paracrine signaling. Paracrine signaling is the sending of a signal molecule from one cell to a neighboring cell. The signal molecule binds to the receptor of a neighboring cell. Examples of intrinsic factors include endothelin, a vasoconstrictor, and nitric oxide, a vasodilator. Both are formed in the endothelial cells of the endothelium of the artery. Another example of an intrinsic factor is myogenic control. Myogenic control is the mechanical activation of calcium channels. This is done by the distension of the artery by the exertion of force from the blood against the artery walls. The opening of calcium channels results in a depolarization of smooth muscle cells, which results in vasoconstriction eventually. So as you can see on this diagram, we have vasoconstriction and vasodilation represented. And in vasoconstriction, there are several factors which cause it, including an increase in myogenic activity, an increase in oxygen, a decrease in carbon dioxide, an increase in endothelin, and an increase in sympathetic stimulation, vasopressin, angiotensin II, and a cold touch. In the case of vasodilation, it is caused by a decrease in myogenic activity, a decrease in oxygen, an increase in carbon dioxide, an increase in nitric oxide, and a decrease in sympathetic stimulation, and heat applied or histamine release. We'll talk about extrinsic factors, which are different from intrinsic factors. And as you can imply from the name, extrinsic factors originate from outside the tissue, and extrinsic factors regulates the blood pressure through the regulation of the systemic resistance. Resistance is going to be referring to the resistance of blood flow. Examples of extrinsic factors include hormones such as angiotensin II, which is synthesized by the kidneys, aldosterone, which is synthesized by the adrenal cortex, and vasopressin, which is synthesized by the posterior pituitary gland. Another example of an extrinsic factor is sympathetic nerve innervation. The sympathetic nerves will synthesize and release norepinephrine, and also the adrenal gland will release epinephrine. So most of these, or all of these extrinsic factors mentioned in this slide are going to result in vasoconstriction. But there are some extrinsic factors that do result in vasodilation, but most extrinsic factors do result in vasoconstriction. One example of a extrinsic factor that causes vasodilation is AMP. So next we'll talk about the pathways or the mechanisms in which the intrinsic factors or extrinsic factors act. So this diagram is showing you 3 examples of intrinsic factors which are going to act on the receptors of smooth muscle cells located in the arterial walls. So the endothelial layer is here, the layer that lines the lumen of the artery, and the smooth muscle layer is this thicker layer. So one intrinsic factor is going to be endothelin, which we talked about, which undergoes or which is involved in the endothelin pathway. So endothelin right here is going to bind to either 2 receptors. So endothelin A receptor or endothelin B receptor. When endothelin binds to the endothelin A receptor, which is a GPCR, a G protein coupled receptor, it is going to eventually result in the activation of our effector enzyme, phospholipase C, as you can see here. So phospholipase C is going to be responsible for catalyzing the breakdown of PIP2 into IP3 and DAG. IP3 is a second messenger which will travel to the sarcoplasmic reticulum and open calcium channels so that calcium can enter the sarcoplasm and bind to troponin on the myofibril, which will result in the formation of cross bridges and eventually result in the contraction of the smooth muscle cell. So the contraction of the smooth muscle cell will result in vasoconstriction. And as you can see, there's kind of a legend right here that shows that the red arrows are contractile pathways and the blue arrows are dilatory pathways. So on the other hand, prostacyclin is also produced by the endothelial cells, and prostacyclin actually can or has a lot of target receptors. So one receptor, so prostacyclin right here, one receptor is the IP receptor. So the IP receptor is also a GPCR that has a stimulatory alpha subunit. So when the prostacyclin binds to the IP receptor, what happens eventually is the adenylate cyclase, the effector enzyme, is going to be stimulated, and adenylate cyclase is responsible for converting ATP into CAMP, a second messenger, which is actually going to result in vasodilation. And we will get to the mechanism later on in this presentation, but CAMP is actually going to prevent the phosphorylation of the myosin heads on the myofibrils. So this will result in vasodilation. On the other hand, we have the TP receptor. And when prostacyclin binds to the TP receptor, it's going to activate eventually phospholipase C. And just like we saw before, that is going to result in the opening up of calcium channels. And calcium is going to go into the sarcoplasm and bind to troponin and allow for cross-bridge formation. And that's going to result in vasoconstriction. So last but not least, we have nitric oxide. So nitric oxide is actually not going to bind to a membrane receptor. What it's going to do is it's going to diffuse through the sarcolemma of the smooth muscle cell. So once it does, it's going to bind to a effector enzyme called guanylate cyclase, which is responsible for converting GTP into cGMP. And cGMP is our secondary messenger, which is actually going to activate a protein kinase, which is going to activate another enzyme called myosin light chain phosphatase, which is responsible for dephosphorylating the myosin heads. And that's going to result in no formation of cross bridges and relaxation of the smooth muscle cell and eventual vasodilation of the arteries. So this next diagram is showing you extrinsic control. So our two extrinsic factors are going to be adrenaline or noradrenaline, which are our catecholamines. So there are 3 different receptors on the smooth muscle cell, which is right here. And so these are called adrenergic receptors, and they all have a different effect on the smooth muscle cell. So firstly, we'll talk about the beta adrenergic receptor. The beta adrenergic receptor has a stimulatory alpha subunit, which is part of the G protein. And what the stimulatory alpha subunit does is it upregulates or activates the adenylate cyclase enzyme, which results in the formation of CAMP. And as we saw earlier, CAMP is going to actually result in relaxation of the smooth muscle cells of the arteries, and that's going to result in vasodilation. On the other hand, we have our alpha-2 adrenergic receptor, which has a inhibitory alpha subunit. And so the inhibitory alpha subunit is actually going to inhibit adenylate cyclase, and it's going to prevent the formation of CAMP, and it's going to prevent the relaxation of the smooth muscles. So the opposite happens. The smooth muscle contracts, and that's vasoconstriction. Lastly, we have our alpha-1 adrenergic receptor, which has a alpha-Q subunit, which stimulates a different enzyme, phospholipase C, which is responsible for converting PIP2 into IP3 and DAG. And like we saw earlier, IP3 is a second messenger that's going to travel to the sarcoplasmic reticulum, open those calcium channels, and allow calcium to come into the sarcoplasm and bind to troponin, which is going to result eventually in the formation of cross bridges and smooth muscle contraction and subsequent vasoconstriction. So this is kind of a comprehensive diagram showing you the different factors at work on how they are going to induce either vasoconstriction or vasodilation. So, like we saw earlier, the alpha Q subunit is going to result in the formation of IP3 second messengers, which is going to travel to the sarcoplasmic reticulum right here, SR, and it's going to allow calcium to come out into the sarcoplasm. So calcium is actually going to bind to another protein called calmodulin, and the calcium and calmodulin complex are going to actually be responsible for activating the enzyme myosin light chain kinase, which is responsible for phosphorylating the myosin heads, resulting in contraction of the smooth muscle cell. On the other hand, we have our stimulatory alpha subunit, which is going to result in the increase of CAMP formation. And CAMP is actually going to inhibit myosin light chain kinase and prevent it from phosphorylating the myosin head. So that's going to result in relaxation of the smooth muscle cell and subsequent vasodilation. In regard to the inhibitory alpha subunit, it's going to inhibit adenylate cyclase, and therefore CAMP is not going to be formed, and so myosin light chain kinase can function in uninterrupted and it results in the phosphorylation of the myosin heads and contraction and vasoconstriction. Last but not least, we talked about nitric oxide. And as you can see, nitric oxide does not bind to any cell, the membrane receptors. It just diffuses through the sarcolemma, and eventually it's going to lead to the formation of cGMP. So cGMP is going to actually activate a protein kinase, which is going to activate eventually myosin light chain phosphatase, which actually dephosphorylates this myosin head, and it results in the relaxation of the smooth muscle cell and eventual vasodilation. So after looking at these mechanisms, we can see the reasoning and we can see the process in which the artery is going to either constrict or it's going to dilate. So there are several different mechanisms for each of these factors, intrinsic or extrinsic, that's going to result in our physiological response. So in summary, after this lecture, you learned that there are extrinsic and intrinsic factors that induce vasodilation in the artery or arterioles. And also you learned that there are extrinsic or intrinsic factors that induce vasoconstriction in the artery and arterioles.